Okay, we are now ready to start on our next lab, which is population ecology. Okay, and for this, so this is how species interact with other species, one population interacting with another population. Okay, and that could be interacting with individuals within their own species in their population, right? Like the population of humans, or it could be interacting with a different species, right? And how that works and how species impact each other, right? Are they uh, does one species population grow while the other shrinks, right? So we're looking at those impacts. So some things to keep in mind, and I'm gonna go to a whiteboard here now and start just kind of explaining some of these concepts, right? So in general, populations can grow or shrink, right? So if this is like time here, and this is our population size, a population size can just kind of grow over time, right? But eventually, most populations kind of even out at a certain size, right? They just don't keep growing and growing and growing and taking over the world. Um, and so this like kind of maximum size that most populations reach is their carrying capacity, okay? That's what this is called. So the maximum number of individuals within a habitat of one type of species, okay? And that could vary, right? So the number of squirrels that can happily live on the Montgomery College campus let's say Montgomery College is 20 acres, I have no idea. And maybe 100 squirrels can live there. But you could go to a different 20-acre area that has way more trees and no building, and maybe 1,000 squirrels can live there, right? So the carrying capacity is unique and different in every environment, right? So it can vary based on the resources that are available for the organisms there. If a population gets too big, I'm trying to, again, get to my colors here. Okay, so let's say we're talking about squirrels and the population grows and it gets too big. There are going to be some things that push it down and force it to get smaller. It can't keep growing beyond its carrying capacity, right? So it'll dip lower. If it gets too low, it will have the opportunity to grow. Okay, so populations may go around this. What are some things? Take a minute and think, what are some things that could keep a population from getting too big, that will kind of push it down if it gets too big for its carrying capacity? So think about that for a sec. Pause me. Okay, so some of those things that can affect a population size are the resources, right? So it could be food. It could be, if you're a plant, it could be like sunlight, right? Like access to the sunlight, which is really competition, right? It's competing with other plants, right? That may crowd it out, right? So it's not the sun itself that's keeping it down, it's the competition with other plants. So there may be diseases, right? If too many individuals get in a certain space, right? Diseases spread, kind of like social distancing. If you put 100 people in a classroom, you're not social distancing and you're going to be spreading diseases. If you only put 3 people in that classroom, um, there's less disease getting spread around, right? Predators. If you're a predator, are you going to go to where there's a lot of food available or where there's a little? Predators are going to be drawn to a dense population where there are a lot of individuals, right? So predators will come into this population if it gets too big and kind of bring it down again. So all of these things, this food, competition, and it could be for sunlight, it could be for nesting places, right? It could be for burrowing spots, right? All of those, just competition for resources, diseases, predation, all of those things will kind of come in, and as a population gets too big for its carrying capacity, it can kind of push it down. If you look, food, competition, diseases, predators, these are all living things, right? These are all biotic factors. Bio means life, right? So the biotic factors are density dependent, which means the denser a population gets, the more individuals in a certain space that you have, the stronger those forces get, right? They're really strong as it gets too big. and they kind of get a little bit weaker as it gets lower, right? Then things like abiotic factors, like rain or drought or wind, right? Those, if there's a drought, it doesn't matter whether you have 100 squirrels or 1,000 squirrels, there's not enough water for them, right? If a windstorm comes through, it's going to knock everybody over. So those factors are not are density independent. They're going to affect a dense population the same as a sparse population. So these are density independent. If we're looking, so now as we're looking at how these populations grow, um, the kind of ideal growth, like nothing can hurt me, right, is the biotic potential of a species. Like how big could this grow, right? And so as we're looking at this population, right, it starts off growing, 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 and it could just, if it had an unlimited biotic potential, and its growth rate, it could just use up any resource, and there was no limits, it could just keep growing. But in reality, right, populations have limits, and they have this carrying capacity, this maximum that they can get to within whatever their population is, right? So I just want to look through the lab, make sure I'm covering um, all of the factors that you need as you're looking through it. Right, so your lab manual also gives you this formula, which you don't need to memorize, but just kind of know if you see it. R is the growth rate, okay? And this is pretty obvious. It's the births minus the deaths. If there's more births than deaths, then the population grows. It has a positive growth rate. If there's more deaths than births, the population shrinks and it has a negative growth rate. Okay. So that's just kind of the background information. Now, what we are going to be doing, we've got two different labs that you're doing here, two different like concepts that you're going to be working through. The first one is, let me clear this, is intraspecific competition. So intra means within the same species, right? If you've done intramural sports, right, those are within your school, and intermural would be between schools, right? So this is intraspecific, individuals of the same species. And so for this one, you are going to be looking at samples of little creatures called paramecium. And this, I will tell you, this is a lab you're glad to be doing from home because these things are like impossible to count under the microscope. This is like the hardest thing to do. But anyways, we've got two flasks of paramecium. Okay, flask A and flask B. Okay, and they were started with just one or two paramecium in each flask. They're the same size, and they were given the same amount of water in here. So and the same amount of time to grow. The difference is that flask A was given 2 grams of hay, and flask B was given only 1 gram of hay. Okay, so there's our limiting resource, right? We have limited hay, which is the food source of these, or maybe there's like an algae that grows on the hay that they eat. But this is the basis of their diet, is this hay, okay? So we've limited the amount of hay and then we've let them grow for 2 weeks. And then what you would be doing in lab and what instead you're going to look at on the slides that are provided is you're going to take 5 samples of each. So you're going to do a count of, for A, you would have count 1, 2, 3, 4, and 5, and the number of paramecium. I think you're doing 5. Maybe it's just 3. Let's see. Yep, 5 samples of each. So you would get a drop of water from flask A and you would count the number of paramecium in that drop, and then you'd go get another drop, count the number in that drop. Okay, you do this 5 times. And then you're going to do it for B also, 5 different counts. Okay. And then you are told in your lab, if you're looking at the lab manual, there are 20 drops in a mill, but your sample average was 2 drops. The flask is 100 drops. It goes through all this. And then it just tells you basically multiply by 1,000 to estimate the number in your population. Okay. So what you do is you take your average of 1 through 5. Okay. So I'm just going to do this very easily. I'm going to go 2, 4, 2, 4, 3. And if I add those all up, 6, 4, 2, 4, 3. And if I add those all up, 6 plus 6 is 12 plus 3 is 15. And I divide by 5, right? Because I have 5 samples. My average equals 3. And then you multiply by 1,000. Is 3,000. Okay, so I can say there are 3,000 paramecium in population A, and I do the same for B, and I'm going to say whatever my number is for B. Let's say I've got like 2,000 is my population for B. Whatever that number is, you are assuming that these populations have grown over these 2 weeks that they were being kept in the lab and then stabilized at their carrying capacity. So we are assuming they are at carrying capacity. So that means they're at their maximum. So whatever you estimate the population size at is the carrying capacity, okay? If you had started counting like 3 days after you started this, the populations would still be growing, right? If you're counting anywhere along this curve, but we're assuming we're at this point, it's flat, they're at a stable size, that's our carrying capacity, okay? So now if you look at the slides, here, we've got our intraspecific, okay? So it shows you, first of all, what a paramecium looks like. Okay, so this is what you would be looking at under the microscope. And you've got these 2 flasks of paramecium in the lab, and you would just be putting them under the microscope and looking. And here's where it's much easier for you because you don't have to actually try to count little white specks. You just go through here, count how many. These are your 5 samples for population A. You've got your 5 samples in the next slide for population B, okay? And is that may be all of the slides, okay? So you do that, and obviously, and then you go back to the lab itself. So calculate your averages for each, and then go back into this lab. That we're working on, and right, it has, here's all our procedures. Okay, interspecific competition. Put the number that you counted in A, the number that you counted in B, your averages, and then you estimate. Remember, your estimate is just multiplying by 1,000. It told you to do that, right? It said multiply by 1,000, so just do that. And then don't worry about the class average, okay? Obviously, we don't have a class average here, just put in whatever numbers you have. Okay, then you get into the questions. What are the carrying capacities of A and B? It's whatever you calculated as the population size, okay? What is the variable in this experiment? So I'll tell you what, why don't you take a break, stop this, pause this. I'm going to continue right now. I want you to stop, do the lab, and then come back. If you just listen to me now, you've done nothing and you're not going to learn it and you won't be able to do it on the exam. So just pause for a moment and then come back, okay? All right, we're back. Presumably you have gone through this and filled in all of the information. Okay, so if we were looking at the paramecium slides, for sample 1A, you had 6, right? For 2, you had again, 6. For sample 3, you had 5. For sample 4, you had 5, and for sample 5, you had 4. Okay. So if we add those up, that's 12 plus 10 is 22, plus 4 is 26. Did I do that right? Hopefully I did. They've made this tough. They didn't make it easy math, that's for sure. So you've got 26 divided by 5 is your average. Oh no, I'm sorry, my average is 26 divided by 5. I wrote 26 in there. Our average is 5.2. 5.2. And so our estimated number is 5,200. Okay. 5.2 times 1,000. Okay, and then we go on to population B. Sample 1 has 3. Sample 2 has 3. Sample 3 has 2. Sample 4 has 2, and sample 5 has 2. So this gives us our is 12 individuals total divided by 5 samples, right? So we have 12 divided by 5. And our average of these is 2.4. So our estimate is 2.4 times 1,000, so it's 2,400. Okay, so B had half as much food, and its number is about half as much. It's not perfect. It's not exactly half as much, right? But it's about half as much. So what was the variable in this experiment? What accounts for the differences between population A and population B? I'm looking at number 2 here. It's the amount of food, right? Food or hay was our variable. Our carrying capacities, I'm going back to number 1. For A was 5,200, and for B, it was 2,400, okay? What would you expect if you gave them 4 grams of hay? So that would be giving them twice as much as A had. If you gave it twice as much hay, it's going to be 2 times as big. I'm going to say it's going to be about 10,400, right? 5,200 times 2. If A had 2 grams, if we give it 4 grams, it's going to get doubled. If it only had a half a gram, it's going to be half the size of B. So 0.5 grams are going to get about, it's going to be about 1,200. Okay, if you double the amount of food, you're going to double your population. If you have the food, you'll have the population. Okay, so that is just a quick and dirty way for calculating your carrying capacity. Okay, so that is the end of this portion of the lab. I'm going to stop the video now, and we will do the next portion of the lab. Actually, you know what? We'll just run it on the same video. No need to be separate. Okay, you can pause now if you want, though. No, you know what? I'll pause it so you can take a break and just start fresh with a new recording. We will